Welcome you all to the class of drug discovery and drug designing. So dear students, today we are going to learn about what is drug and what is drug discovery and drug designing process. So before discussing the drug designing process, we will first discuss what is drug and what is drug discovery process. So let's start with drug. What is drug molecule? Because you are the student of pharmacy, you must know what is the drug and you are familiar of this drug. What is drug? Can any one of you define the drug in your words? Yes, Akash, can you define?
Without excipient, the drug has to treat and cure the disease and disorder.
Drug has to treat and cure the disease. Okay. Without
Excipient मीन्स मैम जो टैबलेट में मैम जो टैबलेट होता है बहुत सारे एक्सपेरिमेंट बनाए जाते हैं, लेकिन मैम उसमें मैम एक एमजी का सिर्फ जो मेन ड्रग होता है एक एमजी का वो बहुत हल्का होता है, बहुत कम होता है, बाकी जितने सारे मिलके एक्सपेरिमेंट बना के उसको मर्ज करके टैबलेट पंच करके तब बनाया जाता है. तो विदाउट एक्सपेरिमेंट मैम मीन्स सिर्फ ड्रग केमिकल कंसिडरेट.
Okay, what we call that?
Chemical considered disease to cure the disease and disorder.
Okay, that is a chemical that can cure the disease in your terms. You want to say this. Okay. So here what is written? What is the definition? Drug means particles. Oh, chemical. Okay. Intended for use in the diagnosis. Okay, we can use in the, but cure and treatment is more important, I think. Okay. And it is used in the prevention of disease in man and other animals. We can have some more definition of drug also, some more easy definition. So because you are from the pharmacy background, you can tell me more definition. You know better than me. Okay. So drug is any chemical or biological substance, synthetic or non-synthetic. This is simple definition. Drug is any chemical or biological substance. It can be chemical or biological. Because most of the time we take the natural medicines. Because we eat the food and we sometimes if we feel any problem, we used to take the home remedy. That is also the biological substance. Natural substance we are taking. It may be synthetic or non-synthetic. Non-synthetic. Okay, and when we take the medicine prescribed by the doctor, okay, it can be semi-synthetic also. Okay, there are different kind of medicine. It can be semi-synthetic also. Okay, semi-synthetic means the drug has the natural origin, means it is extracted from the natural plants, herbaceous plants. Okay, then it is processed. We do the processing to convert into the drug. I mean that can be effective against the disease. So you can mix up these two definition. I think you can take the definition that is combined with these two. That will be the best for you. Means drug is any chemical or biological substance. Okay, that can cure the disease. That can be used to cure the disease or used in the prevention of disease. Okay, this is true. So you can combine these two definitions. Okay, now it is easy to define the drug. So drug is any chemical or biological substance, synthetic or non-synthetic. So in the second definition, we have taken the nature of this is focused on the nature of the drug. What is the nature? What is this? That what is the function of this drug? Function you can take from the first definition. There is a function of drug. So first of all, structure is important. What is drug? Any chemical or biological substance, synthetic or non-synthetic. Okay, and it is used to cure the disease, to treat the disease or the prevention of disease. Okay, in man or other animal. Okay. Okay, sources of drug. So there may be the different sources depending on the origin. Means animal. There can be the medicines. Means we can take from the animals like insulin. Insulin is the example. Okay. So insulin is also used for the treatment, and that is taken out from the pig and cow. Growth hormone. Okay. So this can be used in the digitalis purpurea, foxglove. So these are name of papaver somniferum. These are names of plants. And digitalis is obtained from the foxglove. Morphine is obtained from papaver somniferum. Okay, so name is given in the bracket. These are the name of plants. And name of the drugs are digitalis and morphine. What is the source of these drugs? Plants. There can be inorganic drug, arsenic, mercury, lithium. But these molecules also have the side effects. Okay. So inorganic compound is used, but these have the side effects. Okay. Synthetic, chemical. We call them a chemical drug or chemical molecules. Okay. Although everything that we also extract from the plants, they are also the chemical by the nature. I mean, in the science we define them the chemical, but if they have the natural origin, we call them the natural or synthetic that we synthesize in the laboratory. Okay, you do the practical in the lab, you try to synthesize the molecule using the chemical. The name of the chemical drug, the example is propranolol. Okay. Biological is penicillin. The source of the penicillin antibiotic is biological. Okay. Biotechnology, that is the name of brand. This is not the source. Okay. Human insulin, that we also use some technology to isolate the insulin. Although insulin is produced in the nature in our body, no? Okay. Processing in it, but we take out. We isolate the insulin from the human body or other from the animals to treat the human disease or diabetes. Because biotechnological method we use, that is not the source. Okay, so these are all different types of drug. Means you can define in your terms. Whatever you know, how much you know, you can define. Means what is the drug. Now come to now we are going to discuss the drug discovery and development process. What is this? Okay. How we can do this? This is the difficult process. Somebody can find it. So this is a difficult process. So in the figure you can see, one has tried to say that this is a very difficult process. You have to find out a small thing in a big. Okay. So by doing the experiment you can find it, by the serendipity there is one mechanism. By chance you can find it, but it is very hard. This is just impossible. You are lucky if you are luckily find out the drug. Okay. Okay, found. Okay, so somebody has taken the small time to develop the drug, to discover the drug, but there may be the more time. As now it takes lots of time for the experimental studies, for the isolation, experimental studies, preclinical studies, then clinical studies. So it is a long process, and it always takes the billions of dollars. So one need more money. So that's why the cost of any medicine is very high, any chemical medicine. Okay. So how drugs discovered and developed? Occasionally new drugs found by accident. That process, this phenomena, that process is called serendipity. We call that if we accidentally found the drug. Suppose we have the disease and we have one medicine at our home. We have taken that medicine. If I don't have anything other. Why? Okay. Just saying. Okay, there is a network. No network is coming here. CHGB network is coming. Just trying to connect, I think, CS by CSJB network.
इनसे सर्वर थोड़े कभी कभी प्रॉब्लम ज़्यादा करते हैं.
The problem here, huh?
आप हमारे वाले सिलेक्ट करा लीजिए.
Is yours? You have password?
Yes, ma'am.
No, just leave. Just be like that. No, okay, the class is on recording. We will upload this class. Okay? No problem. What is serendipity? Serendipity means finding a drug by chance. Means we have found the drug by chance, by accidentally. Suppose we're trying to discuss. Okay? Means suppose I do not have any option. I got pain in my stomach and I do not have anything. So means whatever I have at my home, I have taken that, to cure. Accidentally it has been cured. I got relief. So by the accident, means you have found out the solution. You have found the solution. Okay? In the lack of proper treatment. So occasionally new drugs found by accident. It happens. It's how the tribal people do. They used to treat their disease. They don't go to the hospitals, but they know how to treat the disease. They do, means occasionally they visit the hospital. They used to cure their disease. They warn deep wounds also. Okay? Serendipity is this man. Understood? What is serendipity? By chance we found the drug. More frequently they are developed as part of an organized effort. Okay? Means lot of people are working to find out the drug. I mean, this is the usual process. What we do in the laboratory, what you do in the pharmaceutical laboratory or pharmaceutical company, they are trying hardly to discover the drug, for the coronavirus also. So this is the organizational effort, organized effort to discover the new ways to treat the specific disease. Means we have to discover, we want to discover the drug, and we also discover the new ways to treat the drug. That is not always the chemical. We use the different kind of therapy. We have the different types. Allopathic medicine, Ayurvedic, or these are different kinds of. We also have another kind of therapies also. Alternative therapies that we use. You can also study about the alternative medicines, alternative therapies. So in some disease they are useful, but in other disease that may not be useful. Okay, so it depends. So we are just discussing. Okay, if there is no disconnection, the class will be on the recording. Okay, we'll upload this. Okay. Drug discovery, one way to discover drugs. Okay, so in this figure you can see, so blindly, I mean the scientist is pouring a chemical in the beaker. Are you able to see the figure? Why? Okay. Here you can see, no? Okay, you can sit here. Okay. Are you able to see here? Yes. Or you can sit there, here on that side. You sit there. Okay, then you will be able to see. Okay. This is. I mean the scientist is trying. So in this figure you can see the scientist is trying blindly. They are pouring chemical into the beakers, and by chance they will be because they are working. So they try their ideas. So we can also take the idea if we have treated any disease at our home, it works on me, so I can test that chemical or anything that I have used for anybody else to cure that pain or anything else. So if it has worked, I can also work on that using those chemicals. I can make using those substances like the tribal people do. They used to take the leaf and they used to chew that leaf. They used to cure their wounds using the leaf. they apply. The leaves of maize also are different on their wounds, and in that way they treat the disease. Okay. Okay. So here you can see, and they are wondering, what he is doing in this figure. But we have to do it. It cost high. Because we are doing the experiment, we are trying in the multiple ways. And we are using the chemical, then we have to test it on the live animals. We have to carry out first of all, we have to carry out the laboratory testing, toxicology testing, then clinical trials. So it takes lot of efforts, lot of money also. Okay. So there's Dr. Arnold Moore, M.Sc, is conducting an experiment to test the theory that most great scientific discoveries were hit on by accident. Okay. So this is it. So we can say that were hit by on by accident, but it doesn't happen at once. We have to try. These things happen those who are trying. Okay, accident will also happen in most of the time. Those who are working. Okay, because they are trying, they are finding the solution, so they will find out the solution. Those who are not working, they don't know what is coming their way. If they have the solution also, they cannot see what they are looking for. So we have some objective, then also we can find out the solution. Drug discovery by serendipity. Okay, in 1928, Fleming studied staph. Okay, but contamination of plates with airborne mold. Actually, he was doing the experiment, but there was the contamination of plates with airborne mold. What he has seen? The bacteria were lodged in the area of mold. Mold product inhibit the growth of bacteria. On the Petri plate, he has seen the clear plug zone, clear zone. So thanks means there is some infection which is killing that mold. So that was because that means then the antibiotic penicillin was discovered. Okay. Okay, he has discovered. So he has seen on the Petri plate, then he isolated that chemical or that what is the source. Okay, then he discovered this penicillin antibiotic. The source of this medicine is biological. Okay. Okay. Development of propranolol. Okay. Propranolol. Okay. Beta blocking. Okay. Have unexpected give a benefit of discover practolol. Okay. So this is the drug. If we do some kind of development or some change in the propranolol drug, so there is a discovery of practolol. Okay. So propranolol is beta blocker, but it is a lipophilic drug and can enter in the means actually what is there is a side effect of this drug, propranolol. Okay, and cause the side effect. How it? By introducing hydrophilic amide group. So now what we have done? Okay. Now we have made some changes in this drug. Okay. Then we have developed another drug that is called practolol. Okay, using the propranolol, we have developed this drug. Okay. So why it causes the CNS? Because it crosses that, it reaches there. Okay, practolol produce more selective cardio-selective beta-1 inhibitor with fewer side effects on CNS. So in the what is this name? In practolol, we do not have the side effect as much is it there in the propranolol. Okay, so what we have done? There is a change, like amide group. Okay, we see we can analyze in the practice what is the structure of this propranolol and what is the structure of practolol. You can see the difference. You can analyze. Sulfonamide and tolbutamide. You can see the difference. So these are the So this is the drug discovery process by the serendipity. The worker in TNT factories always complained from headache due to dilation of brain blood vessels. TNT was the basis to prepare the nitro derivative which were used in angina to dilate coronary blood vessels and elevate pressure. So this is the case we have discussed, that are enough the cases that we have discussed, okay, to understand the serendipity and how we can discover the new drug, okay. And this is the different example, the medicine which is used for the headache, okay. Fact is, worker always complained about the headache due to dilation of blood brain vessels. So TNT was the basis to prepare nitro derivative which were used in angina to dilate coronary blood vessels and alleviate pain, okay. Mustard gas tanks used in the second, where it has been used? In the Second World War, exploded in Italian harbour. It happens accidentally. Sorry, it was used in the Second World War. So they discovered that the person who survived and inhaled this drug lost their defense against microorganisms due to destruction of white blood cells. But this is the side effect of that. We should have the white blood cells. They can also fight. White blood cells. You know what is white blood cells?
Yes,
They work in the immunity. No? They work against the pathogens. Protect. And they are
clotting factor are also important.
They protect from the infection also. They protect us. Okay. So there are different examples. Phenolphthalein was considered a useful dye for cheap wines after a heroic self-experiment. Pharmacologists experienced that it's a drastic direct activity. So there are the different warfarin was used as a rat poison. So now you come to this figure. Here in this you can this is important. You can see the long road to a new medicine. Okay, the process of drug discovery process, it takes a lot of years, 10 to 15 year process. It's not easy to discover a drug, to design a drug. It is a long process. You cannot skip this. Now you have started the process of the corona, but it will take time. Okay, it will take time. But because everybody is suffering, we are facing this problem at the global level, so we are investing too much because our human population is dying because of this infection. Okay, so here you can see in this figure the whole process. This is summarized, whole process summarized, full development of drug, how it happens. We start from the, okay, here you can see registration process, new medicine. This is a long process actually, how the new medicine come, if we want to design a new drug. Registration, we can't do any registration, and it takes a lot of time if you are working. So clinical data analysis, these are the steps. So there are multiple steps. So we test the candidate medicine, tested in the 3 lakh patients. Okay, so large amount of candidate medicine is synthesized. Okay, so this is the phase second. We study in the patient. We use those drugs to study in the patient. They are showing the side effect or not. Volunteers, healthy volunteers, they can also participate, but it is related to safety when the drug has passed like toxicity test or exploratory development. So project team work on that. So large amount of candidate medicine synthesized. So these are the different processes that are connected with each other. Okay, we have to carry out this. There is the synthesis of compound. In the laboratory we so this we all are doing parallelly, but it starts from the laboratory. If the people are suffering from any disease, we start doing experiment in our laboratory and we try to find out the solution for the disease. In the laboratory we work. Okay, then what we can do, there is no drug for the corona, we can screen the already existing drug. Like from the drug bank we take the drug. You just take the drug from the drug bank, then test it against the target. Just look for, if you have the target, just try. Target is there or not, corona target is there or not. So we can screen the drug that exists in the drug bank. Daily we can study 10 drugs or we can screen them. It works. It shows interaction with target or not. Then only it will work. Okay. So we try to that. Screening. So we can use the computer, robotic methods. So we can use the virtual screening methods also. What we do in the computer, that is drug design. And if it needs some modification, we can do. That is called ligand based. That is cheap method. We have already existing drugs in the drug bank. We have database. We already have the drug. What we need? We need the target. We can screen, or we can use also. We can use the on the patient they are dying. We can at least use those chemicals or drugs to test if they are on the last stage of suffering. In the incurable disease, we do that, like HIV or different kinds of disorders. So we used to do that. We test the other drugs. So screening, early safety studies. So we need to take care of this. This is the formulation development. Means we have developed some formulation by doing some studies. We have developed some formulation or the working solutions. Okay, means at the laboratory. Now we know. Now then we can apply on the animals or on the human patients. Initially you cannot apply. You have to do the testing in the laboratory. So there are a lot of ways where you can't use the human or animals. Before that you have a lot of things to do. There are different technology. We can extract the tissues of the human or animals. We can treat those tissues with the drug and we can see what is the side effect. It is not always true. I have sent you a lecture on microarrays. I have also given another lecture on microarrays. Okay, so microarray is the technology. In that we can also do the testing of the drug. Drug is applied with the different, okay, to the samples that is taken from the disease, different stages of the disease. The chemical can work against that or not, can cure the disease or not. So microarray technology is useful, but that is also very costly procedure. Cost is high of microarray technology. Okay, and there is one another branch also that is called pharmacogenomics. In that we consider the genetic profile of the organism or human to treat the disease, to suggest the medicine. There are different drugs. Drug should be given, the disease should be treated according to their genetic profile. Genetic profile is important. Okay. Like Negroes population have different genetic makeup and Caucasian population has different genetic makeup. Okay. So in this process you can see there are a lot of steps and there are a lot of things. Then we can only register the drug. This is the final process. Okay, IND, we file. Company file IND. Introduction of new drug. What is IND? Introduction of new drug. That is long process, it has multiple steps. It is consists of the multiple types of studies, scientific studies, laboratory studies, chemical testing, biological testing, different types of testing. Then only you can file the IND, you can register the drug. So FDA has a lot of process. Okay, then only it can be after the clinical trial, long clinical trial, it is used for the treatment and it is launched in the market as a new drug. So before that we cannot launch. So this is the long road to new medicine. There are the different ways. So we try on the different level at our best, because you are the student, you can work in the laboratory with the chemical. You can take the plant extract, you can test those also. Okay, you can apply in the patient like the plants. With a herbaceous plant, it is means generally there is no side effect because the concentration of that is low. It's very low, lowest. Okay. So there is a difference between drug and extract. Okay, extract is a mixture, but drug is the isolated chemically pure molecules. Potency is high, or efficacy is high. So this is also the different phases of the clinical trials involve high cost and time. Here you can see different compounds is screened. Then there is a preclinical pharmacological testing, then preclinical safety testing, then clinical pharmacological testing. So these are different types of testing. There is a lot of experiment, and cost is also very high. $800 million. Who have that? Who has the courage to make the drug? Discovery. Then there is the discovery, exploratory process. Exploratory development means we can develop the derivatives of the molecule. So the penicillin also, nowadays we have lot of derivatives of penicillin. Like ampicillin is the derivative of penicillin. That is means okay. Then full development phase 3. So these are the different clinical trial also study. So that is the process. Drug discovery approaches. What is it? Serendipity, luck hai. Luckily you have found the solution. Okay. Screening we do. Screening you can do in the laboratory. Means you
in animals.
But in the laboratory you have sample tissues or sample blood, you can try screening. But that is also a very costly process. Screening that I know. I mean the best way you can do it in the computer. Something you can do. Screening, virtual screening you can do in the computer. Before start doing practicals in the laboratory, you can do it in the computer. Let's look what we can do in the computer. We just see what drug can work best. We can do some analysis in the computer. That will reduce the cost too much. That is the use of drug designing. That is the use of bioinformatics. It reduces the cost. Then you say $800 million. So you can reduce. Okay. Screening, chemical modification. Screening is also done in the laboratory. Okay. Like microarray technology, they also wet lab method. Drug is treated, tissue. Sample tissue is treated with the different drugs, and then we see what are the effect. The image of those spots are then scanned and they are analyzed. Have you attended my previous lecture on the microarray? Okay, in that I have taught you how the microarray. So in the microarray we can also make there is a tissue microarray also. Okay, and that are used in the drug testing or analysis of drug, that are important. Okay, so chemical modification, we can do the we can modify the different groups of the same chemical. Okay, on the ortho meta para position. So this analysis we do in the quantitative structure activity relationship. Then we do the rational drug design. What we do in the computer, we use the computational method to design that drug, that is rational drug design. Okay, for the same molecule we can do the changes. You just do the take one drug, then modify the group of that. Using the Marvin sketch you can do it. There are different drug designing and drug software that you can use to design your drug. Just design your molecule, or you can synthesize that in the laboratory also. If it is new or if it is extracted from the plants or any already existing, then you can also modify that in the laboratory. First do it in the computer. Use the computation tool to do that. Then try it in the laboratory. Okay, this is the kind of work that what we do in the drug designing. That is the drug design. Random screening. Screen a large number of synthetic chemical compound or natural product for desired effect. Okay, so as I have already told you, you can use the virtual screening method. What is virtual screening method? We screen lot of compounds against a target. We know the target and we have lot of chemicals, but we don't know which drug will work best against this. So we can screen lot of compounds, thousands of compounds against. Okay, there was one project on the like lifesaver skin saver project. In that they have virtually screened a lot of compounds against the cancer targets. I think in the case of pancreatic cancer. So in that time there was not lot of computers, so they have used the ideal memory of all the computer using the parallel processing. Okay, that they have. So that was the project lifesaver skin saver. Okay, so it is inherently repetitious and time consuming task. It is trial and error approach, but we can use the computer in this. So one doesn't need to know the structure of drug, know the structure of the target upon which the drug will act. Okay, but in the drug designing we need that. If you want to do in computer, then we need that. So but if you want to try, you can, I mean in this way. If you don't want to know about the structure, then you can take the plant extract, you can take the plant leaves, you can eat them, you can use them to treat the disease. But because you are the students of pharmaceutical science, so your interest should be in the structure, function, USCR studies. You can take interest in that. One doesn't need to know about the underlying mechanism of disease. So what the tribal people do? They don't know about what is happening chemically. What process is going on. They know this is the treatment. They have discovered. Because they are trying. Error and trial. They are practically trying. They are applying it. They are learning by the process. You can talk them also. You can discuss with them what are the different solutions, what are the different treatment they take in the different cases, in the different disease. Okay. So chemical modification we can do in the computer, and that is called lead generation natural ligand screen. So first initially we start from natural ligand, then we do the biological testing. So this is the process. Drug design cycle, lead modification. What is lead molecule? That actually work against the target. It doesn't get modified. You can modify anything, but lead molecule should not be. Like if from the penicillin, beta-lactam ring or anything means lead molecule. It should reach the target to work against. If it will get modified, then it doesn't work. It won't work. Drug design cycle, lead modification we do. So you do the modification on the side groups. You change the side groups of the drug molecule by the chemical modification. That's what we do in the drug design in the computer. That is what we do. So in the last practical we have seen that how we should search in the drug bank database. Now we will search for a particular, we will select a drug. Okay. Okay, so this is the process, drug discovery process. Today we have discussed about the introduction of the drug discovery process. What is the drug and what is the drug discovery process. Okay, now you can define the drug. Okay, now you know what is the drug. You already know also, but you can define it in your own ways. And this is the complex study. It consists of design. Drug discovery process consists of lot of steps. Multiple step process, but this thing we can do in the computer. Drug designing we can do in the computer. To reduce the cost, we can facilitate that complex procedure. A lot of money has been invested there, so we can reduce. What we can do in the computer in the drug designing, we can reduce the cost of the experiment. That is a big one. So this kind of project you can do in this subject also. If you have any question, you can ask. Now the lecture is over for today. Okay. I think we have lost the connection. Now we have. sir. They are there or not? I think. Okay, they are all there, I think. So we lost the connection in the class. Okay, no problem. I have recorded the class. Now the class is over. Okay. Okay.